There we are. So I am now going to thank everybody who shared where they're joining us from. Somebody was from sunny Bavaria, so really, yeah, it's great. We've got people really all around the world. And I'm going to hand over and welcome our event chair, who's Professor Nick Jennings, Vice-Provost of Research and Enterprise. And a very big welcome to Nick, and thank you for sharing the time to facilitate the panel today. Over to you, Nick.
Thanks, Nicola. Thank you everyone who's joined us today. It's great to see so many people from so many parts of the world and from our own country. This has been a period of intense research at the college, not only to find a vaccine, but also to advise public and policymakers, understand the disease and create new diagnostic tests. The pandemic has accelerated research in some areas and spurred innovation in others. And we wanted to share with you today some of the exciting research from recipients of the college's COVID-19 Response Fund. These projects help demonstrate the range of important questions that we need to tackle in response to this pandemic. As has been said, the event is open to all alumni and friends of the college. And I'd like to take the opportunity to thank those who've joined us today who are also able to support the fund. I'll say a few words about it, and it will appear on the screen as well. So the fund was launched on the 25th of March in response to the growing need for flexible funding to support new and adaptive research across the college. Since then, over £600,000 has been raised from over 900 alumni, friends, staff, students, and members of the public. The college has also put in £1 million through the President's Excellence Fund. And response fund grants were up to £50,000. This funding supported 42 projects spread across all of the faculties at Imperial College. The research fund then inspired another donor, Community Jameel, to help establish the COVID Excellence Fund, with some match funding from Imperial. This provided an additional £1 million towards rapid COVID research, with larger grants of up to 200,000, and this has funded 10 projects. We found that this type of funding really enabled us to move forward quickly on vaccine research and trials quicker than we would have been able to do so otherwise if we had to go through other routes of getting sources of funding. So today we're pleased to share with you some of the important COVID research happening at the college and to discuss policy and research questions. I'm pleased to introduce our 4 speakers today. So we have Dr. Jethro Herberg, who's a clinical reader in Paediatric infectious disease. Professor Graham Taylor is a professor of human retrovirology. Dr. Beth Holder, who's a lecturer in maternal and fetal health. And Dr. Alex de Figueiredo, who's an academic visitor at the college and also a research fellow at the London School of Hygiene and Tropical Medicine. In a matter of months, they've developed their core research activity to answer key questions that the world is asking about their research. We'll begin with our speakers giving a brief introduction to their projects. Afterwards, we'll take questions from you. So please send in your questions anytime via the Q&A button. So Jethro, let's come to you first. Jethro.
Many thanks. I'm delighted to be here and have an opportunity to speak to the alumni here. And I'm just going to get the screen up. Hello everyone. I'm a paediatrician in infectious diseases and I work at St Mary's campus. And I'm talking about a project that's actually particularly exciting because it's joint with Pantelis Georgiou's team in engineering, and it's the coming together of 2 different teams to bring this project to fruition. And I want to talk about about diagnosis in coronavirus, and I want to explain to you about a project that we're delighted has been funded by the Imperial Experts Fund that we hope will change the way that we can do diagnostics in COVID-19 and in other diseases. And it hangs on the fact that our immune system is an expertly trained diagnostician, and we want to make the most of the fact that our immune system can perhaps make diagnostic tests faster and more accurately than we can, and we want to harness that. So my introductory slide here, I just want to explain to you that the COVID-19 has got many different faces, and so having a diagnostic test that can encompass all of them is particularly tricky. The top part of the screen here shows how if with time in adults with COVID-19, you get a gradual increase in severity in a disease that really changes from an initial phase driven by viral replication associated with the common symptoms that we're all on the lookout for: fever, cough, and loss of smell. And it gradually morphs into something more severe involving the lungs, involving pneumonia. And over time, it's actually the immune system's response that takes over and really causes the damage. So that by the end of on the right-hand side of the screen, you've got a much more severe disease where there may be very little virus actually detectable in the host at that point. And what you're really seeing is the collateral damage from your immune system. And I'm a pediatrician. I'm very interested that the fact that children have a very, very different course of disease with a very mild disease. And it's a really important question to understand why children have such mild disease. I'm going to put that aside for now, but we see children have a mild or even subclinical disease. And then there's a long gap and most of them get better and nothing more happens. But a small proportion of them come back with a disease that is pretty much entirely, we think, driven by the immune system's response in a sort of delayed reaction. And most of those children with this disease, which is called PIMS-TS, and it stands for Paediatric Inflammatory Multi-System Syndrome, temporally associated with the SARS-CoV-2, so you can understand why we have an abbreviation. Most of those children don't have any detectable virus. So they're coming in with actually what's a completely new disease for pediatrics, driven by SARS-CoV-2, but with no virus detectable. So there's a multiplicity of presentations, and each one of these presentations actually is similar to other diseases that we see in adults and children caused by other things. So how can we get the diagnosis right to give the right patient the right treatment? And so we've got a problem that diagnosis of COVID-19 currently is based on virus detection, which is critically an important part of the puzzle, but doesn't address some patients who actually don't have virus present or detectable. And it's also very difficult using a test based on a virus to understand exactly what's going on the host and therefore what treatment to give. So the consequence of this is that our tests often give us a diagnosis late. We miss opportunities for early intervention, which may prevent serious disease. And all of these patients go on antibiotics and only a proportion of them need it. So we're trying to develop a more accurate diagnostic approach for the whole range of COVID-19 presentations. It doesn't depend on detecting the virus, but is instead trying to look at how the host is responding to give us a clue to what we need to treat. And the background to this comes from work that we've been doing in the last decade at Imperial College, is that the different pathogens that we encounter as humans all stimulate the immune system in a slightly different way. So viruses will bind particular types of receptors, bacteria will bind other receptors, and inflammatory conditions, which may have a trigger or may not have an obvious trigger, have also their own independent biochemistry or metabolism, if you like. And the consequence of the recognition by the immune system of viruses and bacteria and other triggers in different ways is that it sets up a different response in the host. And one way we can measure that response is by looking at the genes that are switched on and switched off in the white cells, which are the cells that we can access from a blood sample. And it turns out that a virus illness will switch on a particular set of genes and switch off another set, whereas in bacteria we'll see a complementary set of genes going up or down. And in an inflammatory condition, yet another response determined by another set of genes. And we can distill down from this pattern of gene expression what the key diagnostic genes are for each condition. And so our hypothesis is that every stimulus to the immune system results in a specific response. And if we can measure this, we can work out what's wrong with the patient by going back. And here I want to talk about how we're trying to take this approach and combine it with a really imaginative engineering approach led by Pantelis Georgiou's group in electrical engineering, where they're using those same microchips that you have in your mobile phone or in your computer that are produced in vast quantities and cheaply, but they're using that same type of microchip technology to detect as a detection system for DNA or RNA. And this system uses the same DNA and RNA amplification system, or PCR, that is used in many, many tests, but it actually measures the results in a different way, and that's what gives it the unique advantage. So this is a quick walkthrough of that. You have a microchip embedded on a manifold, and you put your sample on that, and you run a PCR. It's actually a specially designed PCR at constant temperature. And what happens when you have DNA amplification? You actually release a proton with each nucleotide base that's added. And by adapting the microchips, we can use you can use them as an extremely sensitive pH meter, so we can detect the level of target by the rate of change of the pH, rather than much more complicated and long-winded or detection systems we use normally. This is a much more rapid and cheap alternative. And because it's all embedded on a chip, we can actually just simply do this as a mobile technology and result Bluetooth to a phone. So I'm going to play you a video now of the current pilot system, which is called LaceWing, which is that black device in the middle, which is able to measure the key transcripts that we're interested in. And I'm going to start playing this video while I talk over the top. And because what we're trying to do with a simple platform like this is make a cheap, accurate test. We think it can be run in less than 20 minutes with a disposable chip that doesn't require detection of the virus, but instead looks at how the host is responding. And based on the picture of which genes are switched on and which genes are switched off in the host, we can go back and say, aha, this is what's wrong with this patient. And we end up with a geotagged result that can be linked to a mobile platform, which of course is going to be useful in terms of epidemiology and understanding where positive cases are. And finally, because this is a technology which measures nucleic acids, we simply need to change the sequence of the primers to change which targets we're looking at. And that makes this a platform that's adaptable to different types of diseases in the future. So thank you to the Imperial College Jamil COVID Excellence Fund for funding this project, which we will be reporting back on in due course over the year.
Thanks very much, Jethro. So it's a great example, I think, that project of how our engineers and our clinicians really work together to deliver important outcomes that are really central to the pandemic. So our second speaker is Professor Graham Taylor. He's a professor of human retrovirology. Graham, over to you.
Thank you. I'm hopefully able to share my screen, and you can see that. So thank you for the introduction. I'm head of a section of virology, and I don't normally work with respiratory viruses, but we turned our attention to SARS-CoV early in the pandemic to develop a diagnostic test to support testing within our trust. And I'm going to share with you that assay and some of the further research we've been doing in relation to SARS-CoV-2 detection. One of the questions that is often in the public domain, and testing is very topical at the moment, is just how good are the tests that we use? And a figure I often hear is that they detect up to 80% of infection. Now, the reasons why you may not detect infection is because the RNA of the virus is not actually present at the site when you sample it. It may be that the sampling using a swab is not adequately done, and often we're doing self-testing, or that the actual detection process itself is faulty in some way. So when we established our own in-house assay, we added a control for sampling the RNA extraction and the PCR process, and we detect a human RNA at the same time as the viral RNA in each sample. And what we've seen is that provided we can detect the human RNA, the amount of virus we quantify is unaffected by the amount of RNA, but we don't always detect human RNA, and we call those samples inadequate. But in fact, 99% almost of all samples appear to be adequately taken and the processes are working very well. So the remaining difference must be down to intermittent presence of virus at the sampling site. So that's the first point. If you do have a test, and there's 150,000 swabs a day, more or less, being tested in the UK, you'll get a result which says either the virus is detected or not detected. And that leads to an outcome. You'll be asked to isolate, and you'll go down the contact tracing route if it's detected, and you'll be relieved if it's not. But in fact, when you do the PCRs for SARS-CoV-2, the assays are all in some ways quantitative, and you can capture that and translate the signal that you're getting from the viral amplification and add a quantitative number to it. And if you look at this chart, which is just looking at a range of subjects that we've tested by their viral load, you can see it spans about 7 logs. So there's a massive difference in the amount of virus, and therefore the likelihood of people being infectious. And that was one of the questions that we wanted to address with the COVID response funding that we received. And we therefore went to look at a second signal of the virus, the subgenomic or messenger RNA, these shorter RNA sequences, which are found when the virus is replicating, rather than just the total RNA which we use for detection, as a measure that this may be more likely to be infectious virus. And what we were able to show using 2 different parts of the viral genome, the E gene or the N gene, is that there's a very good correlation between the amount of the subgenomic or messenger virus RNA that we see and the total RNA that we quantify in our normal detection assay. Although you will see that some subjects do not have subgenomic viral RNA present, but generally there's a good correlation. We then took the same samples, the virus in viral transport medium, passed them over to our colleagues in Wendy Barclay's lab to do culture. And this is an immunofluorescence technique to detect when virus is replicating in their cell cultures. And we found that there is a correlation between the viral load and also between the subgenomic viral RNA and whether the cultures were positive. So in the highest quartile, 40% of samples were culture positive. And then you can see that we go down with the last quartile, none being detectable. And perhaps usefully, if we couldn't detect this messenger RNA or subgenomic RNA, none of the cultures were positive. So in conclusion, we believe that 98% or more samples are adequately collected and the processing is reliable. There's a massive difference from one person to another as to how much virus they have in their throats, and that is likely to reflect how infectious they are. We can see that by our culture recovery, which is most reliable at high viral load. But if we want a molecular measure of whether somebody's infectious or not, the fact that messenger RNA is not detected when the cultures are negative may be something that we can use to point to when people are no longer infectious without doing culture. There is a caveat, which is when you have symptomatic infection and your viral load is coming down, you're likely to be in recovery and the threshold for being non-infectious may be applied, but there'll be a small number of people that we detect where at the beginning of their infection, before they are symptomatic, where the viral load may be going up and therefore having a low viral copy number may not represent the fact that they will, in a few days' time, be highly infectious. So we just need to bear that in mind. So just a final note of all the people who've been involved in many aspects of this study and our funders, the COVID Response Fund and support from the Dementia Research Institute. Thank you very much.
Thanks, Graham. A really great example of how many of our academics have taken the area and the research that they're doing and pivoted it towards COVID. That's been a key part of this response fund, and it's great to see. So thank you, Graham. So our next speaker is Dr. Beth Holder, who's a lecturer in maternal and fetal health. Beth, over to you.
Thank you. Thank you for the introduction, and it's my pleasure to introduce our project funded by the Response Fund, which is investigating how the pandemic has impacted on maternal vaccination delivery. So maternal vaccination is vaccination that's given in pregnancy. And in the UK, we vaccinate against influenza, which is primarily aimed at protecting mothers who can have very severe influenza infection. And we also offer vaccination against whooping cough. And this is primarily aimed at protecting babies. It was introduced in 2012 following a large increase in the number of cases, particularly infecting young infants. And this resulted in 14 infant deaths. So maternal vaccination is built on the principle that antibody produced in the mother crosses the placenta to the developing baby. So when the baby is born, it has higher levels of protective antibody. But success of maternal vaccination is also built on the principle of vaccine being accepted and taken by pregnant women. And we have been exploring the factors underlying maternal vaccination uptake. And this can be divided into three main themes. That's awareness of the vaccination strategies. So women knowing that whooping cough and influenza vaccination is recommended in every pregnancy. Access to maternal vaccination services, so where the vaccine is delivered, and if pregnant women have the time and the means to attend these appointments. And vaccine acceptance, so women's perception of the risk of the infection you're trying to protect against, and their perception of the risk and benefits of vaccination in pregnancy. And we're now looking at how the coronavirus pandemic has affected these three areas, because we know that the pandemic has had a huge impact on delivery of other types of healthcare. So our study is a collaboration between Imperial College London and London School of Hygiene and Tropical Medicine. It's divided into two main parts. We have an online survey, which is mainly disseminated through social media, where we're seeking women's views on maternal vaccination during the pandemic and how the pandemic has affected their ability to access not just maternal vaccinations, but other antenatal care. We've had 1,000 respondents to date in the 6 weeks we've been running the survey. And then we'll be doing in-depth interviews with a subset of the women who've completed the survey to try and explore women's perceptions of maternal vaccination and how the pandemic has affected their care. And this builds on our previous work, which has looked at both delivery of maternal vaccination and views of maternal vaccination, as well as how the coronavirus pandemic has impacted the ability of people to access childhood vaccinations. We are yet to close the survey when we'll analyse the data, but I just thought I'd show you some preliminary findings. This is the last question we ask in the survey, where we ask whether pregnant women would be willing to have a future vaccine to protect against COVID-19. And we found that 62% of women were likely to accept vaccination during pregnancy. This increased to 81% outside of pregnancy. And 71% of women were likely to accept a COVID-19 vaccine for their babies. So we hope that this study will have impact for patients and society. We hope it will inform best ways to deliver vaccine information during pandemics. So as well as leaflets that are available in hospitals and GPs, perhaps we should think about delivering vaccine information through text messaging, through the post, or through the internet. It will also inform novel ways to deliver vaccines to pregnant women during pandemics. So as well as delivering it in normal healthcare settings, delivering vaccines through pharmacies or walk-in clinics may be a good strategy to ensure good delivery of the vaccine. And also it's aimed to understand how the coronavirus pandemic has affected women's perceptions of the risk and benefits of vaccines and the risk of diseases we're trying to prevent. Thank you.
Thanks very much, Beth. Really interesting and important work, and thank you for sharing those initial results with us on a world exclusive, I'm assuming here. So it's great to see some questions emerging in the Q&A tab. It would be great if I could encourage you to indicate who you would like the question targeted at. In some cases, it's easier to determine than others, but it will just make my job a bit easier in any case. So have a look at the questions. Feel free to upvote those that you would like to see asked, and as I say, if you get a chance to say who they're targeted at, that would be even better. So our final presentation is by Dr. Alex de Figueiredo. Sorry, Alex, I'm going to get it right soon. Who's an academic visitor at the college and also a research fellow at the London School of Hygiene and Tropical Medicine. Alex, over to you.
Thanks, Nick. Good afternoon, everyone. So this study, which I'll talk about today and funded by the Imperial College Response Fund, will be the first study to provide subnational maps and estimates of COVID-19 vaccine uptake across the UK and identify the barriers to uptake. So I'm working with Professor Nick Jennings, who's in the Department of Mathematics, and the National Immunisation Division at Public Health England, in particular, Dr. Michael Edelstein and Kirsten Hurley. Next slide, please. So a vaccine against coronavirus is seen as a silver bullet, but I think if there was one take-home message from this talk, it would be that universal uptake of a COVID-19 vaccine at or above levels required for herd immunity should not be assumed. The UK has quite a long history of vaccine scepticism, and although people in the UK recognise the importance of vaccinating, there are enough doubts over perceived safety issues with vaccines. And these are likely to combine with online misinformation, such as the example in the slide, which was an online rumour that was circulating very shortly after one of the first UK vaccine trials, that one of the first trialists had died after taking the vaccine, which was completely fake news. Sorry, current slide, sorry. Recent surveys estimate that about 60 to 80% of people in the UK are willing to get a COVID-19 vaccine, but only 38 to 50% would definitely accept a vaccine. Estimates of the population that need to be vaccinated to achieve herd immunity are somewhere between 60 and 70%. But these estimates that have been done recently are from surveys that are based on quite small samples. There's also a further issue that uptake of routine immunisations, such as the MMR jab in the UK, are highly heterogeneous across the population. And this is quite problematic. So you need this herd immunity level to be reached across the whole population. If there are pockets of non-vaccinators that form, then they can act as infection hotspots, which not only place that region or those peoples at risk, but connected regions too. And what the effect of that is that if there are pockets of non-vaccinators, then you could have a vaccination coverage rate of 60 or 70% across the UK. But if there are regions, dense regions with less than that, then what that does is serve to increase the herd immunity level required across the rest of the country. So we need 60 to 70% vaccinated, and that needs to be homogenous over the population. A further issue with current surveys is that respondents may overstate the willingness to vaccinate, and this might depend on the time that they think the vaccine will become available. So if you think a vaccine might not be available for 1 to 2 years, then you might be willing to say that you would accept a vaccine. If it's a decision that you had to make tomorrow, then your response might differ. Next slide, please. So there are 3 aims of this project. The first is to estimate uptake of a COVID-19 vaccine subnationally across the UK. So what we want to do is provide county level estimates of the number of people in each UK county who would take a vaccine. We want to identify the determinants of COVID-19 uptake. So in addition to socioeconomic variables like people's age, level of education, gender, we also want to know how knowledge of the pandemic, how their knowledge of the pandemic will affect their uptake choices. Our third aim is to leverage the pandemic to boost perceptions towards routine immunisation. So the UK has had a crash course in epidemiology over the past 9 or so months. And what we want to do is see whether we can use more technical language to boost perceptions towards routine immunisation. So one example of this might be that before lockdown, somebody infected with coronavirus might infect 1 to 3 other people, but somebody with measles would likely infect 10 to 20 other people. And we want to investigate whether presenting that information to respondents would make them more confident in taking routine immunisation, such as the MMR vaccine. Next slide, please. So this is quite a large survey. We're surveying 17,000 people across the UK. It's an online poll, and we're going to sample from minority groups to boost information from groups that are often unrepresented in surveys. The survey is not quite yet in field. There was a final change we had to make, but it will be in field tomorrow. We are monitoring vaccine uptake, as well as the perceived time that people think the vaccine will become available. We will be recording people's socioeconomic and demographic traits, and also attitudes and knowledge to the pandemic to see how those views shape people's uptake choices. The method we'll be using is multi-level regression and post-stratification. I can talk about that a bit later if needed. But if people are familiar with the pre-election polls in the UK for the last general election, this is the tool that was used by YouGov to give constituency level predictions of voting intentions. And that methodology has been used twice successfully in the UK to predict both the hung parliament in 2016, was it? 2017, and also the Conservative majority recently. Next slide, please. So we have some preliminary data on a survey that was conducted in January. This is just showing you that this method works. So we asked respondents, this was before the sort of before COVID, or as it was hitting Europe, I guess. So we asked 4,000 respondents in the UK whether they thought the MMR vaccine was safe. And then we used this tool to map perceptions across the UK. And we found that the percentage of people agreeing that vaccines were safe, or the MMR vaccine was safe, was lower in large urban settings such as London, Birmingham, Liverpool and Manchester. And we found that these perceptions towards the MMR vaccine correlates with MMR coverage, which you can see on the right-hand figure. If you don't use this multi-level regression and post-stratification tool, then you find no correlation. So this provides some evidence, some initial evidence that our method, the method is going to work. Next slide. So there are a number of key research outputs, and these will be delivered through meetings and reports for Public Health England. So we'll be providing county level estimates with an uncertainty range of COVID-19 vaccine across all counties in the UK. So the idea of that is to identify the hesitancy hotspots and intervene through campaign or messaging materials. We want to identify the county-specific determinants and barriers to vaccinating and see if they differ between counties, and again, that will influence the PHE campaign materials. And then we want to establish whether comparative messaging between information about the pandemic and routine immunisations can boost confidence in existing immunisations. And then what we also want to do is prepare for more geographically refined surveys and conduct focus groups in high-risk settings across the UK to understand more specifically what the problems are with vaccinating and how we can best intervene. So thank you very much.
Thanks, Alex. Really interesting stuff. So we have a number of questions coming in on the Q&A tab now. Thank you for those. Keep them coming in. So we're going to go to a couple of those. So a few of those, few near the top of the list, Jethro, are related to you, the work that you presented, I think. So let me just read them out to you. So would the microchip be disposable, or is there a chance of them being reusable? COVID is already proving damaging environmentally due to PPE and the like. Jethro?
Yeah, it's a good question because there's people have highlighted the fact of environmental waste, but also cost. And if it could be reused, it would have a double advantage. In the stage of we're at the moment in development, it is indeed being reused successfully. Now, a separate question would be whether, if you're at the point where you're actually making treatment decisions for a patient, whether you can reuse it and be certain that you haven't got any contamination from the previous sample. I'm not certain about at the moment, but there would be obvious advantages, and and I totally agree with that question that that's something we should be striving towards.
And sort of just relatedly, while we have you on this topic, there's a couple of questions about how it could be repurposed for other diagnostic tests and sort of what would be required in order to do that.
Yeah, so there's a few questions there about about the specificity and about use in other conditions. So just to say a word of background, it's really quite extraordinary, actually, but we find that every viral infection seems to have its own unique signature. As we speak, we've shipped off a bunch of patient samples from children and adults who had coronavirus, and we're getting RNA-seq data back on that, and that'll drive the discovery to choose the transcripts that we'll use on the pilot device. The current situation is that there's publications in the literature of in vitro assays. There's some very limited publications of data to suggest that coronavirus indeed does have a specific signature that distinguishes it from other viral infections. And also there's some data to suggest that the inflammatory response we see in the COVID patients is also can distinguish it from other conditions. So we have every hope that we're going to be able to find some very specific transcripts that typify these particular infection and use them for that. And in the broader question of how we can use it in other things, this work really started off with a comparison of different viral infections in children, and we showed that we could distinguish flu and RSV. And we moved on to show that we could take just 2 transcripts could very accurately discriminate between children with bacterial and viral infection. And absolutely, one of the key aim for our work at the moment is to develop not only that we're testing COVID, but a more general test of bacterial versus viral infection, that if it could be implemented as a cheap, easy to use test that could work on a finger prick sample of blood, for instance, that could really be helpful in a variety of settings, including primary care or in the field in low resource settings. So we're working towards that as well.
Thanks very much, Jethro. So here's a question for you, Beth. It moves when I read it. I was about to read it. Would you expect pregnant women or newborn babies to be priorities for getting a COVID-19 vaccine when available?
That's a really good question. So I think it's coming out that the priority groups for COVID-19 vaccination, is going to be the elderly and those with comorbidities that we've seen to be associated with severe disease. So maybe people with obesity, diabetes, etc. Luckily, it seems that pregnant women don't seem to be presenting with severe disease, which we've seen with other infections. So for instance, influenza, as I said, pregnant women can get very severe disease and even die. The Ebola outbreak as well disease was really, really horrible in pregnant women. And then, of course, things like Zika virus also was devastating for pregnancy. But it seems that the novel coronavirus isn't resulting in either severe infection in pregnancy nor in severe outcome in the babies. That said, I think it is important that we consider including pregnant women in vaccine trials so we're able to assess the safety and efficacy of this vaccine. When it comes to vaccinating babies, I'd be interested to know what Jethro thinks about that. I mean, most young infants and children seem to not have severe disease, but of course we are seeing some syndromes presenting. I guess it depends how long the vaccine is effective in those babies as well.
Jethro, do you want to add your piece?
Yeah, I think the other side of the coin is also that at the moment we're in a very interesting experiment where the schools have gone back, but other parts of society are still remain in some degree of lockdown. And I think children, if it's anything like other respiratory infections, are very expert sharers of infection. And even if it doesn't affect them badly, there are implications for their parents and grandparents. So I think we have to put that into the modeling that is going to be done to work out whether they should be a key group. I wouldn't be surprised if they are indeed a key group.
Thanks very much, Jethro. So a question for you, Graham. Regarding the test and trace system, why do they not use viral load to target contact tracing?
I wish I knew the answer. Been advocating it
Speculate.
for some time. I think there are a number of technical difficulties. The assays which people are using, they, on the whole, although they're quantitative in a sense that you can tell by how quickly you see the product, so how quickly you detect the RNA in the PCR assay. They all have that information, and the quicker you detect it, the more there is to start with. So you've got a semi-quantitative way of detecting it, and they have that information. They've not converted it into numbers like we did, but that's a fairly simple trick. But then that information is not being requested. So if you're part of the test and trace testing system, you're only asked to report detected or not detected, or in some cases weakly detected as well. So all the assays would have to be interpreted slightly differently, but I do think it's doable, and I do think it would be a useful way of focusing attention on people who are most infectious.
Thank you, Graham. We have a question on the potential negative effect of social media on the uptake of vaccines. Perhaps Alex could take that question first, and then also on for Beth as well. Alex.
Sure. I'm not sure how to best mitigate it. That is a tough question that I know that a lot of advocacy groups and social media companies are looking at, how best to target online misinformation and what best to do about it. One of the problems with removing it is that often gets publicized and makes it even bigger news than if you were just to leave it alone. Often you see social media groups at the moment just putting a link to the World Health Organization or some other national health authority for correct information. We're currently looking at the effect, like quantifying the effect that social media or misinformation online can have on people's willingness to vaccinate. And we find that there is an effect, and it does drop people's willingness to vaccinate by a small amount. So maybe it's not as much as you might think, but still a significant fall. So the problem then is how does that tie in with the number of people that you already would have vaccinating? So is that going to be a significant enough drop to not have herd immunity? So identifying where that will be the case, so geographically and in what social groups, is an important question to find an answer to.
And what would be your best guess, if I was to put you on the spot at this point, of the impact of this on any uptake of the vaccine?
Yeah, so it's something that is incredibly hard to measure. We've devised a survey that looks at this, and we find that there's a fall in the number of people who would reply yes, definitely, that they would take a COVID vaccine, of about 5% when they've been exposed to misinformation. But again, that's under specific circumstances within our survey, so it's hard to generalize that. So we think the effect is likely to be reasonably small but significant, but that it depends on how much of the population is already taking the vaccine. If you're in or around that herd immunity level, then that drop can be of much more impact than if you already have 80+% of people taking the vaccine.
Thank you, Alex. Beth, from your side.
I agree, it's a really complicated issue and it's almost impossible to mitigate. I mean, social media particularly is an echo chamber. So when I go on social media, I generally see pro-vaccine stuff, where other people who are less vaccine confident will tend to see more negative messages. I think if we often focus on the risk of the vaccine, that can make people more hesitant. But actually, when we focus on the risk of the disease we're trying to protect against, that can make people reassess their perception of vaccines. And there are some really great groups who are sharing about how dangerous some of the diseases are. So things like Vice-Provost, they're really prolific on social media. I'm talking, this is a family whose young infant died of whooping cough, and the mother wasn't aware of the maternal vaccine. And they've been great advocates for sharing their story and saying how devastating it was for them not to know that there was a vaccine. So I think stories like that, personal stories, can really help people understand why we're offering vaccination in every pregnancy.
Thank you, Beth. So the question that's next bubbled up to the top is from Francis. I might ask it in a slightly more general form, if it would all stay still for a moment. Francis says, my wife was diagnosed weekly positive in Italy, complete nightmare, as it would seem they have detected parts of RNA, not the full virus. What does weekly positive mean? Is it infectious in the absence of symptoms? Graham or Jethro? Graham first, maybe?
Yeah, so this is sometimes called weakly detected, and it generally means that the PCR require multiple cycles, more than sort of normal, for the RNA to be detected, to be amplified. And that would therefore, what you sometimes find is if you go back to the same sample and repeat it, or use a different assay or get another sample, you will not detect the virus. It is likely to mean one of two things. It's either a false positive, because there's done so much PCR, you will get some sort of signal which is non-specific, or it's a true positive in somebody with one of those very, very low copy numbers that I showed on my slide. And we would expect that to be somebody likely to be in recovery, but particularly if they had symptoms beforehand and not infectious. But that's one of the questions we've really tried to address. But a small percentage of reports will be weekly positive or weekly detected.
Thanks very much, Graham. Jethro, I don't know if you have anything to add to that.
I would add, though, that I'd say there's a third category of patient who may have significant illness, but it's due to a immune-mediated illness in response to the virus, which may have actually been sometime before. We know from children coming in with the PIMS-TS illness, also called MISC, if anyone's on this call from the US, that the average time between exposure to coronavirus and developing the illness is about 4 to 6 weeks. And so a few of these patients are still positive for coronavirus, but most of them are not, but they are antibody positive. So we can see that they've had exposure to the virus, even if it's gone. But some of these patients are very sick, and although they don't need antivirals, they do need aggressive care. So I suppose that comes back to the fact that I think the viral detection is a key part of the jigsaw, but it's not the whole answer in this complicated illness, which had many heads like a hydra.
Thank you, Jethro. Next question is for you, Alex. So it's sort of, the gist of it is, it depends a bit when you ask your question about sort of vaccine, about sort of what is known about it, what stage it's at, how well it's been tested, how much confidence you have about it. How do you mitigate and deal with those sort of different situations in your survey and in the results that you will present back from it?
Yeah, sure. So I agree that the answer that you'll get to these type of survey questions will depend on a lot of things. After we ask people whether they would take a COVID-19 vaccine, we go into depth about the reasons that they may not want to take a vaccine. And I think most of the things that you mentioned in the question, Anthony, are posed to respondents. So we ask them whether speed of vaccine development is an issue for them. Do they have safety concerns over the speed of a vaccine that is being developed on a relatively short timescale? We don't provide any assumptions to respondents, although we do try to understand as many factors about their reasoning as possible. As I mentioned earlier, we ask respondents when they think a vaccine will become available, and we think that's an important question to, if you like, weight responses by. So if people think that a vaccine will become available within the next few days, then they're more likely to think seriously about the risks, the risks of vaccinating. So we try to account for as much information like that as possible. I just noticed the question below. Did you want me to answer that? Because
yes, I was seamlessly going to come to that question, Alex. But since you figured it out for yourself, what's the answer?
So just to say that we're not discarding herd immunity. So if that must have come across. If that came across earlier, then that's certainly not the case. So the point that I was trying to make is that even if the whole country had 70% vaccine, so what we're aiming for is 70% vaccination rate, vaccination uptake of a COVID-19 vaccine across all subnational regions, if you like. If you have uptake that is below 70% in any vaccination, in any region in the UK, then what that does is it serves to disproportionately increase the herd immunity level required in areas that are connected, physically connected to that region. So what this means is that, if you like, that depending on the uptake rate and structure across the UK at a subnational level, we might find that herd immunity levels will differ spatially as well. So it might be the case that in London you might need a higher vaccination rate among people that are willing to accept a vaccine to kind of mitigate those who are just unwilling or are going to flat out reject a vaccine. So we do need herd immunity.
Thank you. Beth, I don't know if you have anything further to add to the herd immunity question as well?
The only thing I would add is that we would normally, when we talk about herd immunity, we're normally talking about immunity induced by a vaccine. So ideally, we would want to deliver a vaccine to as many people as possible to achieve herd immunity without anyone actually suffering from infection and disease. And I think sometimes in the media and in politics, herd immunity is being used to talk about infections instead. So making sure that as many people as possible can be infected and thereby achieving herd immunity. But ideally, we'd want to achieve herd immunity through vaccination without anyone getting sick or dying.
Okay, thank you, Beth. I want to come to a couple of more general questions for the panel to consider now as we sort of come towards the end. So I'm going to come to each of you in turn, in the order in which you spoke, just so as you're all prepared for this. So what do you think it's going to take to get us out the other side of this pandemic, both from the kind of work that you've spoken about and perhaps more generally? Jethro.
Thanks, Nick. I think that high quality data, epidemiological data, and intelligent interpretation of that data to give us sensible public health measures has to be a cornerstone of what we need to do. But alongside that, I think if there's a small silver lining in the situation, it's catalyzed lots of groups to turn their attention to COVID, to take on slightly different tasks. And we really need to embrace the cutting edge technology that's coming out of this. And that includes diagnostics, which is where I work. It also includes the treatments. I mean, there's been incredible rapid response and development of new clinical trials on the fly. And we also of course, there's all the stuff that's going on in vaccines. So I think it's public health plus all these other bits. It's not a single magic bullet.
Thank you, Jethro. Graham.
Sorry, I've muted. So I think at the end of the day, we are either looking at needing the vaccine or an effective treatment for the virus, because at the moment, our strategy is simply protecting people from infection whilst we find a way to tackle this pandemic comprehensively. The coronavirus is not going to go away. So lockdowns are temporary measures. Isolation is going to reduce transmission, but unless we have that herd immunity that we've been talking about, we're still going to be facing coronavirus problems in the future. If the vaccine, and I expect the vaccine to be delivered and to work, but were it not, then I think we would be looking at antiviral therapy, particularly for people most susceptible, as the next sort of target to get us out of this.
Okay, thanks very much, Graham. Beth.
I agree with both the previous responses. So I guess from my perspective, I think something that's really important that we need to sort of expand on is looking at how we can deliver other healthcare during the pandemic. So thinking of, as I said, novel ways that we can sort of restore some kind of normality to other healthcare, you know, like treatment of cancers, and we know vaccination rates have gone down. So I think that's something that will need to be developed as we move forward until vaccine is available.
Thank you, Beth. Alex, from your side.
Yeah, probably just parroting some of the previous responses.
The trouble of going last, Alex.
So yeah, we do need a safe and effective vaccine, and we need a high willingness to vaccinate. And I think that important in developing vaccination community strategies will be looking at subnational patterns and trying to boost immunisation rates in areas with low rates and high vaccine hesitancy.
Thank you, Alex. So I'm going to come back to the audience questions in a moment, but I wanted to ask another one of my own. At some point, hopefully, this pandemic will get past us and we will think about future pandemics and making sure that we are better prepared as a country and as a world to what those might look like. So how do you think sort of these experiences that we're having at the moment will help us effectively respond to future pandemics? And I'm going to go in reverse order this time. So you can get to say something first, Alex, without having to think of an answer and then a new answer.
So I think a major issue is trust in government and trust in authorities. So we've seen that trust in the UK government has fallen quite substantially since the pandemic started here. But trust in both government and health authorities is crucial to for uptake in not just vaccinations but other instructions like lockdown that the government might give you as well. So yeah, I think that maintaining high confidence in vaccines is important as well, and being able to convince people or administer other health services in a safe way as well that people have faith in. So as Beth said earlier, we've noticed that vaccination rates for routine immunisations have dropped across the UK. We know that those who are at risk of developing serious side effects from seasonal influenza are also in a similar bucket to COVID as well. So we need to make sure that, for example, when administering other vaccines like the flu vaccine, that they feel safe and comfortable getting the vaccine, and that might mean other modes of delivery, such as home vaccinating or something like that. So I think we need to consider quite carefully how we maintain high quality healthcare in other areas that might be shut off during lockdowns. Thanks
very much, Alex. I think that's brilliant. I would agree with that very much. Beth.
So I think a lot of the public health strategies and messages that we have been going out will really help us to respond to future pandemics. And I think it's shown us that people are really interested to understand the disease and how it works. And actually giving people real information helps them understand why we're asking them to make the changes to their life. And now people understand words like PPE and viral load is part of an everyday normal conversation. It's amazing.
Who'd have guessed? Who'd have guessed? Thank you, Beth. Graham.
I think what we've learned is that you need a strong public health system, and our public health system has been gradually sort of taking less importance. And I think that's partly because we thought that infections were not as important in the present, millennium as they were in past millennia, and that's almost certainly true. But I think this has shown us that infections are very important and we need to be ready for them. The other thing I would hope we would have is a much better homegrown industry to provide, so we can do our own development of testing strategies, reagent supply, whether that be PPE or the molecular techniques or even therapies. I think we need to build in those areas.
Thanks, Graham. And Jethro.
I agree with everything that's been said, and so I just want to pick up particularly on the diagnostics, because that's where what I've been talking about today. And I think that one thing that we've learned is that we need to have really strong diagnostic capacity across the UK to cope with large numbers, and we need to have imaginative testing strategies. And I think although we haven't solved this problem in its entirety yet by any means, the headlines every day are still talking about the problems of getting enough tests done. I think the development of new testing strategies that can be adapted quickly to new pathogens is really central to having that ready in your armory against future pandemics if it's a new pathogen coming along. So I'm hoping that we are actually, through the pain we're going through, we are shoring up our defences for the future at the same time.
Thank you, Jethro. I'm going to go back to, we've got about 5 minutes left. I'm going to go back to some audience related questions now. And Jethro, I think the first one is for you. Can the handheld equipment be used as a quick test to discriminate between viral and bacterial infections in GP surgeries to reduce things like overprescription of antibiotics?
Well, I touched on this earlier, and the answer is that we hope it will be able to do so. And in fact, the work we're doing now on coronavirus comes on the back of a project that we were already running to develop a test exactly like this that we could roll out in primary care. And actually particularly close to our heart is to try and get it rolled out in low middle-income countries as well. So, because we think this can be it doesn't require a lot of complicated equipment. All you need is a mobile phone, which is really ubiquitous. So we're hoping we can develop this kit more easily for that bacterial and viral question, which is very important, than for a lot of other questions, because we know the difference in people's response in their gene expression is really clear cut between a bacterial infection and a viral infection. There are lots of nuances which I won't get into now, but the short answer is yes, we hope so, and we're working on it.
Great, thanks very much. Next question is around the speed with which we are producing vaccine for COVID, which is on one level a fantastic achievement of really getting people to operate at unprecedented speeds, but actually in terms of sort of public confidence might give the impression that some corners are being cut. So how can we, are there previous examples or case studies in engaging more with the public to help improve their confidence in vaccine programs, particularly when perhaps they've moved at pace? So I might come to Alex first, and then Beth, I don't know if you have a view on that as well. Alex.
So I'm not aware of previous public engagement strategies. Maybe Beth has something to say about that. But yeah, I can stress that is a concern that we are picking up in our surveys, and something that we are monitoring. We know that in the US, I think there is, the amount of people that there's a significant fall in the amount of people that would take a vaccine if it was available before election day. So it's obviously a major concern, and it's something that we're monitoring. And yeah, I'm not sure I can speak further to previous case studies.
Thank you, Beth. Anything further to add to that?
I also don't really have specific case studies. I would say generally when a new vaccine is introduced, there may be lower uptake. So we saw this with maternal vaccination. When we started our study in 2012, when the whooping cough vaccine was first introduced, uptake of flu vaccination was much higher, and confidence in flu vaccination was much higher because people were more familiar with it. And we generally find people are quite, they take into account the views of their family members, their friends. So this can also be a big impact on whether people are willing to have vaccines. So I think it might just take time as people become more used to the idea of being vaccinated.
Thanks. Jethro, I think there's a couple of sort of specific diagnostic test questions for you coming up around sensitivity, which I think you've touched a bit upon, but you might like to elaborate. And then sort of in practice, as in when we get out of the lab into real world operation, what's the likely sort of false positive rate for these sorts of technologies? Perhaps both in terms of the ones that you're developing, but sort of maybe answer it more broadly as well.
So the sensitivity seems to be very good, and a number of experiments are being done at the moment to try and go down to the very lowest number of copies of target to see what it can be picked up by the test. And actually it looks like it really is just down to a handful. So if your sample's got even a handful, in this case of transcripts, then the test can pick it up. What the test is aiming to do is to be quantitative, because what we're comparing is the difference in gene expression between two different conditions. And it's not an all or nothing. It's about how much you've got. When you have a viral infection, you upregulate your interferon stimulated genes. So you've got more copies of the messenger RNA molecule. And so we need to detect the numbers. And but the range of that the assay can detect seems to go down very low, which is very important. In the real world, what's the false positive rate going to be? I think we don't know the answer to that yet. And until we've got a device that is close to being ready for market use, it will be hard to say. But in the design of the research we're doing, one of the things we'll be doing is bringing these machines into healthcare settings, generating results on patient samples in real time and comparing them to the results that are generated by other conventional tests. And so at the first stage of that, we won't be using those results to influence patient care, but one of the key questions will be, how often does it get it wrong? And so that's something we'll need to look at closely.
So thank you very much, everyone. I'm going to draw this panel to a close. I would like to thank all the speakers for their really insightful contributions, and the audience for some great questions that have really sort of enlightened the discussion, I think. So really very much appreciated. It's great to see Imperial's science and research really at the forefront of helping with this, our country's response to this pandemic. And we've seen 4 great examples of that going on today. So thank you very much, everyone. And back to you, Nicola.
Thank you, Nick. And thank you, everyone. That was just as Nick was saying, just such an amazing sharing of both research and insights, but also just such a range of questions around public health policy to very technical ones, which were probably beyond me, but for some of them. But I just want to thank, we're very lucky at the college as well, just to have a great sense of community and collaboration. And it takes time to put on these events for the researchers to go out their valuable research time and to prepare and to participate. So a big thank you to all of you, very grateful to that. And I just want to thank you again for joining us from around the world, and keep safe, keep well, and we'll see you at our next event. Goodbye.